Hi, this is Kim Osborne. I've been privileged to work under Annie Ice and Dr. Ian L. Popa as they strive to develop a procedure for measuring the interaction between an antibody and its antigen using a fluorescent marker. Antibodies are Y-shaped proteins created by the immune system. They are tasked with the job of identifying and eliminating substances the body doesn't recognize. Regions at both tips of an antibody are hypervariable and can conform to specific molecular structures found on the outsides of pathogens. Certain bacteria defend themselves against this by expressing proteins on their cell surfaces that bind to mammalian antibodies. One such protein, expressed by certain members of the species Peptostreptococcus magnus, is known as protein L. It binds with high specific affinity to an antibody's variable region within the kappa light chain domain Fab, in such a way that the antibody can still function as normal. Those qualities are why our lab chose protein L to take our mouse IgG1 antibodies. We desired to improve upon current methods of measuring the interaction between an antibody and its antigen using a fluorescent marker. This is currently done with the Western blot, where protein of interest is loaded onto a gel and then transferred to a membrane. The membrane is then incubated in its first set of antibodies, which selectively target the protein of interest. Next, the membrane is incubated in a second set of antibodies. These antibodies carry luminescence, and they seek out the primary antibodies. The luminescence allows researchers to visually quantify the attachment of the first set of antibodies to the protein of interest. The method we developed both simplified and standardized this Western blotting quantification process. We began our assay by directly electrophoresing a stable concentration of mouse IgG1 antibodies onto a blot. Functionally, this behaved as a protein of interest incubating in its primary antibody. Our next step was to slice the blot, then incubate it in 10 different concentrations of a protein construct meGFPL, which is protein L covalently bound to the green fluorescent protein meGFP. We imaged the blot under a photosensor to quantify our results, and a typical example blot is pictured here. We drew horizontal lines through the fluorescent IgG1 antibody bands to obtain the relative intensities. The same was done with the loading control so its relative intensity could be found and used to normalize the antibody fluorescence. To see how well protein L bound to the antibodies, this normalized data then needed to be analyzed through the lens of a dissociation binding constant known as Kd. Kd is defined as a concentration of ligand that occupies half the receptors at equilibrium. If a Kd value is small, the affinity is strong, as only a small concentration of ligand is required to occupy the receptors. Conversely, if a Kd value is large, the affinity is weak, as a larger concentration of ligand is required to occupy the receptors. This can be represented by the following equation. Fractional occupancy equals the concentration of the ligand divided by the quantity Kd plus the concentration of the ligand. When we plotted our normalized data using the Kd equation, our Kd was revealed to be roughly 30 nanomolars. This is a small value, meaning our construct meGFPL had a strong affinity for mouse IgG1 antibodies. Our method of using fluorescent proteins for labeling, rather than two sets of antibodies as in Western blotting, has many benefits. It allows for faster exposure times of well less than a minute. Its detection sensitivity includes the nanomolar and micromolar ranges. The bioluminescent signal is more stable than the chemiluminescent signal typically used in Western blotting. Finally, the fluorescent protein does not interfere with the functioning of the antibody. To conclude, our method shows great promise in medical and research applications through its simplification and streamlining of the Western blotting process.